Okay, just another couple of things to mention. Another way to culture bacteria or to create a bacterial lawn would be to use a plastic L-shaped disposable spreader. So obviously it comes sterile in a packet. So you open the packet, you dip it into the bacterial solution, you open the Petri dish just a little, and then you gently spread the spreader over the surface. When you have done that, you then pop the spreader into disinfectant so that the bacterial culture does not contaminate the environment. So you have closed your Petri dish. How do you label Petri dishes? The label obviously should be on the outside, and the label should be on the base, not on the top of the Petri dish. The Petri dishes should be incubated upside down. Why is that? Because water evaporates from the agar jelly. So if the jelly is on the top, when the water evaporates, it falls away from the agar jelly. So by incubating it upside down, it stops the condensation falling on the agar, and that would destroy the bacterial culture. So that's really important. And then, as I mentioned in the last video, you incubate at a suitable temperature, but it should not be 37 degrees C. 37 degrees C would encourage the growth of human pathogens. So this is one of the experiments that you could be asked about, the effect of antibiotics on bacteria. So you start with an agar plate which is covered with a bacterial lawn. So in other words, the agar has been inoculated with bacteria. And then onto the bacterial lawn, you put 2 little antibiotic discs. So in this experiment, we have 2 different antibiotics, antibiotic 1 and antibiotic 2, and you incubate the plate for 24 hours. And then if around the antibiotic disc there is a clear zone, that means no bacteria can grow in there. So what has happened is the antibiotic has diffused from the little disc through the agar and it kills the bacteria. Now, if you look at this, the clear zone around antibiotic 1 is much larger than the clear zone around antibiotic 2, and so antibiotic 1 is more effective at killing those bacteria. Now, just a couple of things that come up. When you're putting the discs on the plate, they should be far apart so that the zones do not actually overlap. And unfortunately, whenever you go to measure the clear zones, measuring the diameter is quite difficult because the clear zones can be uneven. So it's best to measure the radius a number of times, and that will give you more accurate results. Another thing is the discs should not be placed too near the edge of the plate, because that would mean that you're missing the clear zone on the right hand side. So the ideal is that the clear zones do not overlap, or that the discs are not at the edge of the plate, and then you're missing half of the clear zone. And remember, the radius is a more accurate, taking lots of radial measurements is a lot more accurate than measuring the diameter. Now, here is another experiment that you could be asked about: antimicrobial plant properties. So some plants are really good at killing microorganisms. So many plants release chemicals as they grow, and they do that to defend themselves from attack by bacteria and fungi. So how do we get these antimicrobial chemicals out of the plants? You grind the plant up in a mortar and pestle in water, and that releases the cell contents into the water. Then you would take some sterilised tiny little filter discs and you would soak them in that plant extract, and then you would add the discs to an agar plate which has a bacterial lawn or a fungal lawn, very like the experiment I just talked about. You would seal the plate and you would incubate that plate for 24 to 48 hours. You would take it out and then you would look for clear zones around the little discs. So when we did the experiment, we found there were large clear zones around the garlic discs. So something in the garlic had antimicrobial properties, had diffused out of the disc and had killed the bacteria around the disc. So the clear zone does indicate that the extract, the plant extract, has actually killed the bacteria or fungi on that Petri dish. Now, it's really important in this experiment to have a control. So a control is really just a disc that you have soaked in water, and it does not have any plant solution on it. And that proves that it was the plant extract that caused the clear zone. Because if you soak a little disc in water and you put it in the dish and you see the clear zone has been caused by the water and not by the plant properties. So that's why you need to do control, and it proves that it is the plant extracts that are killing the bacteria and nothing else. The last thing then I want to talk about are E-strips. E-strips are a sophisticated way to measure the effect of antibiotic concentrations. So you're trying to work out how effective an antibiotic is. So an E-strip is actually a strip which has a concentration gradient of antibiotic along the E-strip. So down here there's a very low concentration of antibiotic, and the concentration then increases, and at this end of the E-strip you've got a very high concentration of antibiotic. So the E-strip again is placed on a Petri dish which has been inoculated with bacteria. It will be incubated, and then whenever you come and look at it, you'll see a pattern like this. So this concentration of antibiotic is no good. It does not kill the bacteria. And then we can see that this concentration of antibiotic, it does kill the bacteria, but that is not as effective as this concentration of antibiotic. It kills a lot more bacteria. So what is the point of this? It makes it easy for people in the labs to determine the effectiveness of different antibiotic strengths. There's no point giving somebody a really, really strong antibiotic that could have really, serious side effects. It can make them feel quite ill. If you could use a lower concentration of the antibiotic, get pretty much the same effect, but there would be fewer side effects. And there's no point giving somebody an antibiotic of a strength that we know would not kill the bacteria. Okay, so again, I want you to look at your past paper questions, look at the mark schemes, and make sure you can answer all questions on this topic.